Hi, welcome back to my channel. In this video, we will be talking about the citric acid cycle. So, citric acid cycle was given by Sir Hans Krebs. It is a strictly aerobic. It is a strictly aerobic reaction. It occurs in mitochondrial matrix. All right, now let's go through the pathway so that it is easy for us to understand. So first of all, we have pyruvate from glycolysis. This pyruvate converts into acetyl CoA through the reaction of PDH complex. Now this complex has 5 components you should know about. One is FAD, then we have NAD, then we have CoA enzyme, then we have lipoic acid, and we have DPP. Now this acetyl CoA combines with the oxaloacetate to form something which is called citrate. All right, through an enzyme called as citrate synthase. All right, now this citrate will further form cis-aconitate. Now cis-aconitate will form isocitrate. Isocitrate will form oxalosuccinate. Oxalosuccinate forms alpha ketoglutarate. Alpha ketoglutarate forms succinyl CoA. Succinyl CoA forms succinate. Then succinate forms fumarate, and fumarate forms malate, which in turn forms oxaloacetate. Now let's just talk about the enzymes. So over here we have the enzyme as aconitase, here also aconitase. Then we have isocitrate dehydrogenase, here also isocitrate dehydrogenase. Then here we have alpha ketoglutarate dehydrogenase. Here you have to remember enzyme which is succinate thiokinase. Here the enzyme is succinate dehydrogenase. Here the enzyme is fumarate dehydrogenase, or wait, here the enzyme is fumarase. So here the enzyme is fumarase, and here we have malate dehydrogenase. Now I will tell you some key points. So let's just discuss about the energetics. So in energetics, you have to remember that here we get NAD, FAD, and GTP. Alright, first of all, you have to remember that through succinate thiokinase, we get 2 GTPs, which further get converted into 2 ATPs. Okay, then I think this color is that much peeling. So 2 GTP is converted into 2 ATP in the step of succinate thiokinase. Then, so this is through the 2 GTP through succinate thiokinase. Then we have this step over here. In this step we get NAD. So we get 2 NAD here, then 2 NAD here, then 2 NAD here. Alright, so 3, right? So we have around 1, 2, 3, 6 NADs. So we get them through the alpha ketoglutarate dehydrogenase, then malate dehydrogenase, and isocitrate dehydrogenase. So 6 into 2.5. So this is basically, even if you see, if we so this forms 15 ATP. All right. Then so this is 2 ATP. Then we are still left with FAD. So FAD is through succinate dehydrogenase. So through this step you get 2 FAD. So 2 FAD, so 2 into 1.5, so we have 3 ATP, right, through the step of succinate dehydrogenase. Alright, now you have to remember one more thing. In alpha ketoglutarate, the reaction before it also, like CO2 is given out, and the reaction after it also CO2 is given out. Alright, remember this. Then you have to remember that this step of succinate thiokinase is the example of substrate level phosphorylation. Okay, now pathway we have done. Substrate level phosphorylation is through the succinate thiokinase step. This question, major anaplerotic reaction, मतलब which reaction is giving the intermediates for the cycle? It is the pyruvate carboxylase enzyme. Now this reaction is basically nothing but pyruvate getting converted into oxaloacetate inside the mitochondrial matrix. Then vitamins involved in TCA are vitamin B1, B2, B3, B5. Energetics I have done, and now just let's go through the activators and inhibitors. So basically the inhibitors, so enzyme and the inhibitor, right? So just enzyme and inhibitor. Alright, so we'll talk about the first we'll talk about aconidase, second alpha ketoglutarate dehydrogenase, and then third is succinate dehydrogenase. So aconidase is inhibited by fluoroacetate, alpha ketoglutarate dehydrogenase through arsenite, and succinate dehydrogenase through malonate. So if here you see total, so 15 plus 3 plus 2, so you have 20 ATP being formed through the TCA cycle. So this is the TCA cycle. If you like the video, do subscribe to my channel. Thanks for watching.